Assalamu alaikum dear students. Welcome to Dr. Roomi lectures. In this lecture, we will study the regulation of thyroid hormone secretion. In order to maintain the normal metabolic activity in our cells, it's important to have a steady rate of secretion of thyroid hormones so that we maintain a very precise level of thyroid hormones, which actually is done in our body. Feedback mechanism operates between hypothalamus-pituitary and thyroid axis. This controls the secretion of T3 and T4 hormones. The most important regulator of thyroid hormone secretion is thyroid stimulating hormone TSH. Normally there are no wide swings in the secretion of TSH, which means it is very finely and precisely regulated, and then it controls the levels of thyroid hormones within a narrow range. The secretion of thyroid stimulating hormone or thyrotropin from the anterior pituitary is increased by a hormone, thyrotropin releasing hormone, which is released from hypothalamus. A nucleus in hypothalamus called as paraventricular nucleus is stimulated by low temperatures and then increase the production of TRH, which stimulates anterior pituitary to increase the production of thyroid stimulating hormone. It has been observed experimentally in rats that when they are exposed to low temperatures, there is increase in production of thyrotropin releasing hormone, which increases their body metabolism up to 50%. In human beings, newborn babies, when are exposed to low environmental temperature as compared to the warmer intrauterine maternal environment, there is increase in production of thyrotropin releasing hormone, and this increases the production of TSH hormone from the anterior pituitary. This is important to increase the release of T3 and T4 hormone from thyroid gland so that there is increase in body metabolism and increase in heat production, which is important for their survival. However, in adult human beings, this has not been observed to increase the production of TRH when we are exposed to cold temperatures. However, when adult persons are exposed to very low temperatures, for example, in the Arctic environment, and we go and live there for several months, then the secretion of TRH hormone is increased, which increases production of TSH and then thyroid hormones, and this increases the body metabolism up to 15 to 20%. Anxiety and excitement because increase the production of catecholamine hormones in the body and also heat production in the body. These two factors inhibit the production of thyrotropin releasing hormone from the hypothalamus so that there is less production of TSH and then thyroid hormones, and there is no further heat production in the body. Prolonged fasting, starvation, and stress of some infections and chronic diseases on the body inhibits the release of thyrotropin releasing hormone. And one mechanism for this is decrease in production of leptin hormone, which decreases the production of TRH, so that there is less production of TSH and less production of thyroid hormones. This may be important to conserve the calories of the body. TRH is a tripeptide hormone which is released from the paraventricular nucleus and then comes into the median eminence of hypothalamus. Then it is absorbed into the blood vessels of hypophyseal portal circulation, and then along those vessels it reaches to the anterior pituitary to stimulate the basophilic cells in the interior pituitary, which are called as thyrotrophs. TRH acts on its receptors on the thyrotrophs, and then second messenger mechanism inside the cell is activated. This is a phospholipid second messenger system which increases the production of inositol triphosphate, diacylglycerol, and calcium, and in this way it increases the production of thyroid stimulating hormone from the thyrotrophs of anterior pituitary. If in a disease the blood vessels of hypophyseal portal circulation are blocked, then TRH will not reach to the anterior pituitary, and then the secretion of TSH hormone from the anterior pituitary will be decreased. Thyroid stimulating hormone is a glycoprotein which acts on its receptors on the thyroid follicular cells. Because it's a protein hormone, it cannot cross the cell membranes. There is some second messenger mechanism here which increases the production of cyclic AMP. This increase in cyclic AMP production stimulates various steps of thyroid hormone synthesis. Thyroid stimulating hormone has quick and delayed actions on the thyroid gland. Very quickly, it increases the uptake and proteolysis of the thyroglobulin and then release of T3 and T4 hormone into the blood circulation. It also increases the uptake of iodide, iodination, oxidation, and coupling reaction of the thyroid hormone synthesis. The long-term effects of thyroid stimulating hormone on the thyroid gland is to increase the number of cells and also the size of thyroid gland, which means it causes hyperplasia and hypertrophy of the thyroid gland. It also increases the blood supply of thyroid gland. In case of increased production of TSH hormone, there is increase in the size of thyroid follicular cells which change from cuboidal normal shape to columnar shape, and also there is increase in the number of cells and also size of the tissues. But in case of decreased production of the thyroid stimulating hormone, there is decrease in the size of cells, which means cells become flat instead of being cuboidal, and also there is atrophy of the thyroid gland. T3 and T4 hormones, after they are released, they travel through blood circulation to their target organs. They increase the metabolism in our tissues. T4, which is more abundant in our blood circulation, is slowly converted into T3, and T3 is more active biologically. Both these T3 and T4 hormones have negative feedback effect on hypothalamus and on anterior pituitary, but their negative feedback effect on anterior pituitary is much more than their effects on the hypothalamus. It is the free form of T3 and T4 hormones, not the bound form, which have negative feedback effect on anterior pituitary and hypothalamus. And out of these two hormones, free T3 and free T4, it is mainly free T4 which has negative feedback effect on the anterior pituitary. And this is why as long as free T4 level is normal, even if free T3 level is decreased, there is less effect on TSH secretion from the anterior pituitary. For day-to-day regulation of thyroid hormone secretion, free T3 and T4 have the negative feedback effect on anterior pituitary to control the secretion of thyroid stimulating hormone. In anterior pituitary, T4 hormone is converted into T3 by the action of deiodinase enzyme. This T3 at anterior pituitary decreases the receptors for TRH, and thus by downregulating the receptors of TRH on thyroid troughs, it decreases the sensitivity of thyroid troughs to TRH. Here are some other factors which affect the secretion of thyroid stimulating hormone. Dopamine and somatostatin, they show inhibitory effect and thus decrease the secretion of TSH hormone. Estrogen has stimulatory effect on the secretion of TSH. It's because estrogen increases the production of thyroxine binding globulin, which increases the bound form, and the free form is transiently decreased. When there is transient decrease in the free form of the T3 and T4 hormones, this increases the negative feedback production of thyroid stimulating hormone. If large doses of iodide is given to a person, this has inhibitory effect on the synthesis of thyroid hormones, which is called as Wolff-Chaikoff effect. And because there is decrease in production of T3 T4 hormone, this increases the negative feedback production of TSH hormone from the anterior pituitary. The effect of free T3 and T4 hormones on hypothalamus is important for long range adjustments of thyroid hormone secretion. We have discussed before that exposure to cold in newborn babies and in experimental animals increases the production of TRH hormone, and these factors, anxiety, excitement, and prolonged fasting, starvation, and infections, decrease the production of TRH hormone and thus the production of TSH and then thyroid hormones. An interesting phenomena called low T3 syndrome is noted in some persons having prolonged fasting, starvation, serious infection, prolonged illness, or after surgery. In these persons, an enzyme called as 5' deiodinase is inhibited in skeletal muscles, and hence T4 is not converted into T3. Because the T3 level is low, which is biologically more active, hence there is less thyroid hormone activity at the tissue level. So less oxygen is used and there is decrease in basal metabolic rate. But because this enzyme is not inhibited in brain, So the oxygen consumption and the metabolic level in our brain remains normal in those people who are having low T3 syndrome. As we have discussed before, decreased T3 has less negative feedback effect on anterior pituitary. So the level of T4 hormone and also TSH hormone remains normal. Now we will talk about a clinical condition in which the size of thyroid gland is increased. We call it goiter. In Pakistan, we call it gillard. Goitrogens are all those chemicals which block the synthesis of T3 and T4 hormones, and then by increasing the synthesis of TSH hormone, it increases the size of thyroid gland. Goiter does not define the functional status of thyroid gland. Goiter may be present in those who are having hypothyroidism, hyperthyroidism, or having euthyroid functional status. Just to mention some important inhibitory factors which can decrease the synthesis of T3 and T4 hormones and hence can increase the production of TSH and increase the size of thyroid gland. Perchlorate and thiocyanate are anines which inhibit the uptake of iodide by the iodide pump at the basolateral membrane of thyroid follicular cells. Thioamides are some drugs which can be used for the treatment of hyperthyroidism. They include propylthiouracil and methimazole. They can suppress the peroxidase enzyme and hence all the reactions which are performed by peroxidase enzyme like oxidation, iodination, and coupling reaction can be inhibited and they can decrease the synthesis of T3 T4 hormones. Increased concentration of iodide in thyroid gland has inhibitory effect on the T3 T4 hormone synthesis and this is called as Wolff-Chagoff effect. In case of iodide deficiency, iodine will not be available for iodination reaction and all these factors can decrease the production of T3 T4 hormone and hence increasing the production of TSH by negative feedback level will increase the size of thyroid gland leading to goiter. So just to summarize the regulation of thyroid hormone synthesis, this is feedback regulation between hypothalamus, anterior pituitary and thyroid gland. Paraventricular nucleus of hypothalamus releases TRH, which is a tripeptide hormone. It has its receptors on the thyrotrophs of the anterior pituitary, which is stimulated by the phospholipid second messenger mechanism. These cells secrete TSH hormone, which has its receptors on the thyroid follicular cells, which are stimulated by the cyclic AMP second messenger system, and hence all the steps of thyroid hormone synthesis are stimulated. And moreover, there is increase in blood supply and also increase in the cell size and cell number in the thyroid gland because of stimulation of the thyroid gland by TSH hormone. Free T3 and T4 hormones have their negative feedback effect both on hypothalamus and on anterior pituitary, but more so on the anterior pituitary. The free T4 levels are more important in the negative feedback regulation as compared to free T3 levels. And negative feedback effect of T3 and T4 hormones on the anterior pituitary is more important for day-to-day regulation of the thyroid hormone secretion. And negative feedback effect of free T3 and T4 hormones on the hypothalamus is more important for long-range adjustments of the thyroid hormone secretion. So this is all about the regulation of thyroid hormone secretion. Thank you for watching this video. See you next time with another video.